Hello, my name is Sylvia Masueda. I live in Dallas, Texas, but I was born in Bogota, Colombia, in South America. So Spanish is my first language, and I actually work as an interpreter. I have two sons, 25 and 20 years old, and my youngest has CF. I am a parent representative on the Cystic Fibrosis Foundation Genetic Therapy Working Group, and will be moderating tonight's webinar. I have been waiting to hear about genetic therapies for 20 years since my son was diagnosed with CF. This webinar will help everybody better understand genetic therapies and their role in CF. Welcome to everybody who's joining tonight, and to those who will be joining us to watch the recording later on. So I'm going to start with some housekeeping notes. We're recording this webinar. It'll be posted on YouTube, and everybody who registered for tonight's webinar will get an email with the recording. We have included time at the end for questions. So if please type your questions in the Q&A box. Please do not ask questions about individual or personal situations, therapies, but we do encourage you after tonight's webinar to have a conversation with your care team about topics, and if you have more questions. There is a chat also in the that is the default is to panelists only. So if you can switch that in a drop-down menu to all panelists and attendees, your comments can be shared with everybody who's attending. And we also do have closed captioning available. You can turn it on or off by clicking the CC icon at the bottom of your screen. So we're going to start with a presentation by Dr. Solomon tonight. He's an associate professor of medicine at the University of Alabama, Birmingham. He is a physician scientist who studies CFTR function and means to treat CF. He is the recent past director of the University of Birmingham Adult CF Center, and now directs the CF Clinical Trials Unit at that university. So welcome, and we can get started.
Thank you, Sylvia, for the kind introduction, and to all of you for being present tonight, and for those that will be watching on the recording for this very important topic. Before I begin, I'll just mention that I have a few financial disclosures that are listed here. However, these will not affect the data or the discussion we'll have tonight. So the portion that I'll be talking about with you for the next few minutes is really centered upon two concepts. The first is understanding the science of the CFTR protein. The CFTR protein is the central protein which has effects in CF and is the causative issue behind the overlying condition. And so we want to understand, how does the body make the CFTR protein, and why does CFTR genes matter in this whole process, and where do they fit into the whole process of making proteins? And so the first section of this will delve into different ways of us understanding those and answering those two questions. First, I want to remind everyone that CF is a hereditable condition. Many of you that are parents or families or people living with CF will be aware of this. But what that means is that CF happens because you inherit a gene, a CFTR mutation, which is an abnormality or an error in the genetic code, which encodes for a protein from both your mother and your father. And by this, the genetics of the condition are what define the overlying and underlying issues which cause CF, including the protein defect and dysfunction, which belies most of the ways that the CF manifests in people living with this condition. And so this slide is meant to remind us that genetics are central in the condition, both for causing the disease and also understanding the effects of the genetic defects which are present, also called mutations. So we wanted to answer the question, how does CFTR protein get to the correct place in the cell to do its job? The correct place in the cells that line the organs in our body, especially the lung and sinuses as an example, is the tops of the ciliated and other airway epithelial cells, which line those those tubes in the lungs and help to sweep mucus out. And so CF goes through a complicated process, that protein that begins with the process called transcription, which takes the genetic code known as DNA into a blueprint copy of that known as the message, or mRNA. Then outside of the cell nucleus, as shown here, a process called translation happens, which takes the blueprint copy, the mRNA or message RNA, and converts it and fully lengthens and constructs a protein in these building block machines called ribosomes. And then the protein begins to fold. After the folding process happens, there are special vesicles that take the protein for post-translational modifications, which basically make it proper and fully folded and adorned with the right kind of things to make it go to the cell surface to do its job. And in the case of the CFTR protein, that job is to move anions and regulate the movement of water across that cell surface to help properly hydrate the mucus layer there and to keep we call homeostasis, which means that we're keeping a happy, healthy environment for the cells in the lungs and other organs in which this protein is being expressed and used. So after those post-translational modifications, if the protein is all okay, that protein goes to the cell surface and does its job. Now let's think again and let's form some analogies that can help us understand the role in which genes, which are essential for building proteins, help to maintain normal health. The analogy we're going to use here, and we'll extend for an analogy we hope will be both easy to remember for you about the function of the CFTR protein and also memorable for you, is that the DNA or the gene is really the hard drive or hardwired copy of a blueprint. The blueprint is a set of instructions for building a protein. The body needs to be able to make multiple copies of that blueprint so that it can make many proteins from one initial blueprint. And that photocopy of the blueprint is what's known as the mRNA or messenger RNA. And it's the photocopy of the blueprint that's taken by the builders, those ribosomes we talked about earlier, to the building site in the cell to make the protein. The ribosome, or the builders here shown in this analogy, then use these blueprints as instructions to assemble the building blocks to build a protein. And in the analogy we're going to carry throughout the rest of this portion of the talk, that protein is constructing a gate. The gate, in this case, is the CFTR protein, which allows the anions, the chloride ions, and water to move through it. And in this case, proteins can be machines or other structures that perform functions in the body, either structural or mechanical or both. And in the case of CF disease, the absence of that CFTR protein, in this case, the absence of a properly functioning gate in a fence, for instance, makes the inability for things to move through the gate like it's supposed to, in this case, anions. And so keep that in mind as we're going through the next few slides, which will extend that analogy in a way to help us understand the therapies which are available for treating protein defects in CF currently. So let's think about our blueprints and gates again related to the types of mutations. So that will give us a framework for how we treat CF. The first type of mutations, known as class 1 mutations, have blueprints which don't essentially call for making any gates. So the blueprint itself is so faulty that no gates are made from it at all. It has that much of an error. The class 2 mutations, and the most common of these is the F508del mutation, which many on the call will be familiar because it's the most common mutation encountered in people with CF across the world. This mutation causes the blueprint to call for only a few faulty functioning gates. And so there are some gates made, but they don't function properly. Now, in contrast, the class 3 mutations do function as the proper number of gates, essentially, where they're supposed to be, in this case, the cell surface for CF. But like in the case of the G551D mutation, this mutation results in a gate that is locked shut. And similarly, the class 4 mutations cause a gate which can open, but it has a block, like shown here with the unhappy face, blocking the movement of things through the gate properly, even though it will open. And finally, we have mutations like the class 5 mutations, which cause the blueprint to make a fewer number of gates than it should. So whereas there may be functional gates to some extent, those number of gates are so few, we can't do the job that the gates are there to do, in this case, the CFTR protein. And in contrast, not having CF or being the normal state of health means that an appropriate amount of gates that open normally are constructed via this process from the blueprint to the blueprint copy through the builders of the protein of the ribosomes and then the function of the gates themselves. So you may be asking, why are we talking so much about gates? Well, let's get into that just real quick. Okay, so in CF, the goal of the gate or the CFTR protein is to move anions, in this case, chloride ions, across the cell surface to hydrate mucus and regulate the health of the cell mucus layer and the tops of the cells in the lung and other organs. Now, in the case of the G551D mutation we talked about earlier, that's a class 3 mutation in which the gates can't open, okay? So we can give a drug known as a potentiator. In this case, the approved one of those is a drug called Kalydeco or ivacaftor. And when we give a potentiator to these locked gates, it acts as a key and opens those gates up and allows now our cows, who represent the chloride ions and the proper movement of things through the CFTR gates, to be able to move from where they are now over to a very green pasture and have a lush amount of grass to eat and be happy cows. So as you can see, with that potentiator on board, our cows can now pass through the previously locked shut gate and move across where they're supposed to, in this case, through the fence, through the now open gates, the cell surface, and over to the greener pasture where they want to be happy cows. Now, let's think now about the F508del mutation situation. We said that has a couple of issues associated with it. The main issue with the F508del mutation is the blueprint calls for the production of faulty gates. So the gates are not constructed properly in this case. And in the case of the actual protein, it means they're misfolded, okay? And so the body tells them not to put those gates in the fence like it should, in this case, the cell surface. And so as a result, we have a fence that totally obstructs our cows from getting across the cell surface or the fence over to this greener pasture. And as a result, we have to think of ways to treat that. So what we can do is give a combination of therapies. And the one that many of you are familiar with is the Trikafta drug, which has 3 drugs in it, 2 of which are corrector drugs, known as alexacaftor and lumacaftor, and tezacaftor are examples of that. The 2 that are in Trikafta are alexacaftor and tezacaftor. And then we can give a second drug, so to act as a key. We'll talk about that in just a moment. So in the case of the corrector, we're basically repositioning the builder to make a properly constructed gate in this case. That's what the corrector drugs do. So they insert a nearly functional gate into the cell surface by pushing that through the production process of making the gates. In the case of F508del, some of those gates don't open properly, and so they respond to the addition of another drug, the potentiator or Kalydeco again, which helps to really open those gates a bunch and allow, in combination with the correctors, for those cows, again, to get across their greener pastures and do the function of the CFTR protein. And so because F508del is apparent in many patients with CF, many patients now have a very effective or somewhat effective therapy to work on at least one of their mutations. But that doesn't account for everyone in CF, as we're aware. And it leaves out 10+% in some populations of people with CF. Many more than 10% of them don't have access to highly effective drugs. So that brings us, to the next question, how do we develop treatments and cures for all? And in thinking about that, how are these CFTR modulator drugs we've just been talking about different from genetic therapies? And what are the types of genetic therapies? And at that point, I'll leave it off and turn it over to our next speaker. So let's think again about our cow analogy and think about the fact that some mutations cause the blueprints not to make any gates. An example of that is the premature termination codon mutations, or the X mutations, like W1282X, a common example of that. And these proteins don't make any protein. And sorry, these mutations don't make any protein. So therefore, we can't treat with the drug that makes the protein itself work better at the level of the builder or at the level of the gate in the fence. So therefore, other strategies will have to be undertaken to correct these types of mutations. And that's what we're going to be talking about the next few moments. So what about gene therapy? So the concept of gene therapy is where we go back in the process, back to the blueprint, or almost back to the blueprint in that process, and replace or fix that blueprint. So in other words, we edit or we give a new copy of the blueprint into the genes with a plan that builds gates that can open easily. So by that, we're basically replacing, in this case, or editing the blueprint for building CFTR way back at the gene level. And that goes all the way to making fully functional number and properly opening gates through which our happy cows can pass through like they should. So it skips the concept of the modulators and goes right to fixing and making appropriately functioning protein, in this case, the gates in the cell surface or the fence in this analogy. So because this is so complicated, the CF Foundation has embarked upon a remarkable concept known as the path to a cure. And through many routes, we'll reach one mission, which is how can we treat everyone with CF at the basic level? And basically what we have to figure out is how can we repair CFTR proteins? That's the case of modulators, which we've talked about a good bit. And there are other modulators being developed to help other people with CF, either more function being gained or more mutations. Can we restore the CFTR protein? Or ultimately, can we fix or replace the CFTR gene known as gene therapy? Let's think about the ways in which we can do this in a simplified diagram. So in the case of having dysfunctional CFTR gene or a CFTR gene mutation, we can either restore the protein function or we can correct the gene. The gene correction can happen by either delivering the new gene, that's known as gene therapy, okay, or fixing the gene, that's known as gene editing. So in the case of gene therapy, we don't worry about the faulty blueprint or the blueprint copy. We don't worry about that at all. We just say, let's just give a new blueprint into our genetic makeup. In the gene editing concept, we would go in and, like a spell check in our computer, and actually correct the errors in the blueprint itself and permanently correct those. Now we're a ways away from ever getting to trials on gene editing. It's much more complicated. So gene therapy or delivering new gene is much more nascent and more likely to happen in the near future for trials. So we're going to focus mostly on that throughout the rest of this discussion. In addition to correcting the gene or restoring the gene, we can restore protein function by either delivering or correcting the faulty blueprint copy known as the message RNA, so giving mRNA therapy, much like the gene therapy delivering the new gene, or we can fix the mutation in ways. And one of those is the concept of read-through, which are drugs which are under development in the early stages, which would fix mutations like the X mutations, like the W1282X and other mutations like that, by actually reading through the defect in the mRNA copy being produced, you can actually make some functional protein. And so with that, I'm going to pause and introduce Dr. Deepika Polineni next, and she will tell us a good bit about the clinical trial updates. And I will say thank you for listening for this portion of the discussion.
Thank you, Marty. My name is Deepika Polineni, and I'm an associate professor of medicine at the University of Kansas Medical Center's CF Center. We can go to the next slide. The following relationships are the research relationships that I wanted to disclose with the commitment, of course, that I will not discuss off-label or investigational use of therapies in my presentation today. Next slide. So one of the most important points that Dr. Solomon and I really wanted to impart from today's discussion is that many people have had the misconception that CFTR modulators are part of genetic therapies, or what we are commonly referring to as gene therapies. But it's very important to note that CFTR modulators are not part of what we are talking about in gene therapy discussions. Dr. Solomon did a terrific job of explaining how the CFTR channel works and how modulators are working to correct abnormal CFTR protein that is being made. But as you've just learned, that's after the genetic code has already allowed for some protein production. In those cases, the protein has been faulty and modulators are able to affect the abnormal CFTR protein. So these are drugs that are being taken orally right now. And the three main types, as discussed by Dr. Solomon, are potentiators, correctors, and amplifiers. But these are not targeting the underlying CFTR gene defect. Next slide. So for the community today, as we talk about Genetic Therapies 101, we're really focused on treatments that are aimed at fixing or replacing the CFTR gene. Genetic therapies and genetic-based therapies are strategies that are going to allow us to achieve CFTR restoration for all people with CF, regardless of their mutations, and eventually a one-time cure. But it's important to emphasize eventually versus what's immediately on the horizon. So as we are looking at genetic-based therapies, we are looking at what's around the corner, and that includes RNA therapies, or dealing with the photocopy of the blueprint, as Dr. Solomon discussed, gene delivery, or going in and delivering a blueprint itself, but gene editing, which many people hear about as CRISPR or similar types of technologies for gene editing, that would still be quite some ways off, and we do not look at that as a treatment or a trial that will be coming immediately in the pipeline. Next slide. Ultimately, we are looking at 4 strategies to treat CF when we group these into different categories. There's addressing the DNA defect, and that is, again, the blueprint itself. That is gene delivery, which we are focusing on right now, and gene editing coming down the way in the future. RNA, or the photocopy of the blueprint, and supplying that into the cell. Those are RNA therapies or short nucleotides, and that's the photocopy of the blueprint as mentioned. When we talk about correcting the protein, we're no longer really talking about genetic-based therapies, because we're talking about the protein that the gene has already coded for. And those are currently being addressed by some CFTR modulators, with more modulators still in research and development. And then, of course, we have the traditional therapies that this community knows so well that have been targeting the end effects or treatment, the manifestations in the organs that are affected by CF. Next slide. So when we think about which therapies are possibly en route as we are moving forward, there is quite a bit in the landscape coming into the future here. The slide is by no means meant for anyone to memorize all of these names. In fact, I hope that the overwhelmingness of this visually is actually encouraging, because this presents a snapshot of all of the activity that's going on in the research community at this time. So we're looking all the way in the left column here at treatments that are targeted towards CFTR restoration. Again, this is really focused on that photocopy of the blueprint and delivering that into the cell. That can be done through mRNA or oligos, and mechanisms by addressing a delivery for pertinent for nonsense mutations and more. And when we go to the second column, we're looking at gene delivery. In this case, we're talking about delivering the blueprint itself. That can be done through a viral vector, so delivering the new CFTR gene in through a virus, or non-viral carriers as well. And once again, for gene editing, such as CRISPR, we're really looking at probably in the next decade, but not immediately in the next years. And still other models and tools are being researched for how we can continue to improve these aspects of delivery. And finally, there is also investment from the CF Foundation and venture capital to keep looking at new research and technologies as they are becoming developed. Next slide. So when we talk about the path to a cure and what we can start to expect in the very near future, we know that we're already looking at CFTR modulators up here in the top right, and further development and research for modulators that may be applicable to additional mutations than what is currently covered. But we're absolutely currently looking at the restoration of CFTR protein or delivery of RNA. And these are 2 groups that are already looking at current preclinical studies and plans for clinical trials coming down the pike, Arcturus and RECODE. And finally, we're also looking at replacing the CFTR gene or delivering the CFTR gene with companies that are planning for clinical trials and conducting preclinical work at this time, including 4DMT, Spirovant, and Crystal Bio. And we do have Orfixit here, but again, we're looking towards that in the future. Next slide. One of the most important points that we want to emphasize today is that we're looking at safety as a community and where we've come from and what we've learned from the past. In the 1990s, after the discovery of the CFTR gene in 1989, there was a lot of hope and enthusiasm in the CFTR community that if we found the gene, then we can find a way to fix it. And we recognize that it's been a long time between then and now for so many people with CF and families that have been waiting. But that's all the more reason why we're so enthusiastic to talk to you all today and answer questions, because we know that we're in a new chapter that is here and just around the corner coming. In the past, we can look at approximately 18 clinical trials with experience, and all of these were really focused on gene transfer through 3 different types of vectors or carriers of the CFTR gene getting delivered into the cell. That was commonly delivered through lipids, adenoviral vectors, that's similar to the cold virus that you commonly think of as a common cold, or adeno-associated viral vectors. So those are kind of cousins or related to the adenovirus itself. But we learned a lot from those experiences. So some of the common themes are that these were generally early phase studies, and they were mostly single center. Well, today we're really looking at taking advantage of what we know on how to build research across the therapeutic development network in the CF community and looking at multi-center trials to be able to enroll people around the country with CF. Industry was mostly not involved in the past, but CFF has done an amazing job at developing partnerships with these industry groups so that the research is really informed by experts in the CF community 360. And in the past, this was also focused on nasal and sinus cells with some lung dosing, but today we're really predominantly looking at delivery to the lungs based on lessons learned from the past. Next slide. So when we look at the past, what were the key results and limitations? Well, one of the first things that we do when we're measuring success is understanding how safe and tolerable a new therapy is. So we knew that we had experience in the past seeing inflammatory responses with safety and tolerability, and we know that we're going to be looking for that carefully as we move into next clinical trials. When it comes to efficacy or how well a treatment is working, of course, we're looking at FEV1, but we also are looking at a number of other biomarkers that we are just starting to be able to detect with higher sensitivity, or being able to get a much closer look at through new technologies that have been developed to understand if CFTR is being expressed and is functioning in the cells. So many of you are familiar with that FEV1 from the blow spirometry maneuvers in clinic. And we know that many people are also tired of the importance that is weighted on that. And we want to emphasize that as we look at these trials, we are interested, of course, in seeing how lungs are improving, but we are also interested in looking first at very careful detail of what is happening as CFTR is being introduced back into these cells. Next slide. So what happens to make sure that we're ensuring safety? The important thing to take away from this very text-heavy slide is understanding that the measures to ensure safety for participants in research are so multiply layered and start before a study has even begun enrollment. So ranging all the way from the approval of the Food and Drug Administration to even clear a study to be able to be conducted. The study is then also being tracked before enrollment and during the study by the sponsor themselves, monitoring medical events with independent medical experts. The Therapeutic Development Network is engaged in looking at the safety and the potential risks of the trial even before it has commenced. And there is a rating that is given from the Therapeutic Development Network, which is comprised by the review of many research coordinators, doctors and nurses that are participating on CF research at centers around the country, and leaders of CF research in our nation at the TDN. And then there's ongoing oversight from the Therapeutic Development Network during the study itself, which is also being contributed to very carefully and rigorously by data safety monitoring boards from the CF Foundation that include experts that have been looking at safety at these clinical trials for years, and that continues to be ongoing. Those are all layers that are higher than what may directly seem to touch a study participant as they start looking at enrollment at their own center, where, or a center that they've been referred to, where an institutional review board has already reviewed and approved the study after what may be several iterations or rounds of discussion to make sure that the study is safe for participants. On top of that, the study team, which is really going to be made up of your doctors and nurses and research coordinators, has to have decided that that study is a study that they want to conduct at their center, that they feel is good for their patients, and they will continue to monitor you throughout the study. And then, of course, I think the most important thing to take away from this slide is that the most important thing is the patient's role. Research is an autonomous decision. So the respect and the rights of an individual participant's research come first before everything to us. You should ask all of your questions, not only during the informed consent process, but as you continue through the study. Next slide. So who is going to be eligible for these early trials? Well, certainly we know that people with CF are the ones that we are aiming for with these therapies. And the first priority is going to be directed towards people who are not taking CFTR modulators. Industry sponsors will ultimately decide who is eligible for these trials in general, and there will also be consideration absolutely for people who are taking CFTR modulators, as gene therapy may have further effects. When we also think about healthy volunteers or CFTR carriers, for example, a parent of someone with CF, we know that they may have some role in participating in early phase 1 studies when we know that we're looking at safety, which is really the important point to emphasize, that the earliest studies or phase 1 studies are looking at safety first, simply to make sure that the treatment is safe and tolerable in the human system. Later studies, phases 2 and 3, then go on to look at the measures of how effective these therapies are. And absolutely in those stages, we will be looking towards people with CF to enroll in these studies so that we can find new treatments that work for them and for all. Next slide. So how can you find out about these trials? Trial Finder is a website that we would absolutely direct you to. That there you can filter the studies by status, by location, age, and more. Here you can see that you can even put in your zip code and start to find trials that are within a distance that you can pre-specify as far as how far you are willing to travel for a study. But do contact research coordinators at your center for a study that you're interested in, or just to learn more about these trials in general. And you can sign up for email alerts to be notified of enrolling trials and new study results. And you can use this QR code also to help you get to the CFF.org's trial finders. Next slide. So in summary, what we are really discussing today are 3 groups of therapies: RNA delivery, looking at the photocopy of the blueprint and getting that into the cell; gene delivery, or the blueprint itself; and gene editing, which is really taking the construction all the way back to the beginning and rebuilding that blueprint to start. The third of these will be a long ways off, so we'll start with the very top. For mRNA delivery, yes, we do think that all people with CF will be eligible, but again, the priority is going first to people who are not on CFTR modulator therapies from what we see. Dosing schedules can be anticipated to be weekly, and we're expecting that these will be delivered to the lungs at first. And there are potential challenges, of course, that any cell that takes it up will make protein, and that is part of why we're looking at the lungs at first, but also trying to get into very specific cells in the lungs that are really depending on making that CFTR protein well. It's not a permanent cure. And we're also looking at gene delivery or gene transfer, again, eligible for all people with CF. We expect that this may be monthly. This is also being targeted towards delivery to the lungs at first, and with the same challenges that we're really trying to get these into specific cells in the lungs that depend on making CFTR to function properly. But what will be a permanent cure one day, we expect, will be gene editing. We don't know what that dosing schedule will look like. We think that it will be systemic delivery, and we know that it will be able one day to edit DNA outside of the CFTR gene. And that is part of what is taking so much longer to get into considerations of that, because there is so much safety to consider there. So we can move to the next slide. We just want to emphasize that we know how diverse and important every member of the CF community is, and that our discussion today is really focused on therapies that can meet the needs of all people with CF. These trials we know will not move forward without your participation, which really should happen with your questions answered and your safety always taken into the first consideration. So we'll move to the next slide and give you a resource here for where you can direct further questions before we move into our Q&A. So you can search for enrolling trials, one more time, clinical trial finder. You can ask your care team or contact research at cff.org. And you can always check out further reading on genetic therapies on cff.org. And here's an example of that page showing researchers in CFF laboratories and expert scientists and physicians around the nation who are working on these strategies today. Thank you so much, and I look forward to rejoining Dr. Solomon and Sylvia Masuera right now so that we can answer your questions.
Thank you so much, Dr. Solomon. I loved your analogy of the cows and the gates. It's such a complicated topic, and it really makes it easier to understand. And thank you, Dr. Polineni. It's encouraging to see so many trials in that slide. It's kind of coming near finally, and we're kind of moving forward. So we are going to start our Q&A portion of this webinar. I would like to remind everybody, you can type your questions in the Q&A icon at the bottom. And I think we're ready to get started. So this first question is for Dr. Polineni. When will clinical trials for genetic therapies start?
So this is definitely an exciting time in CF genetic therapies and clinical trials. We're already expecting to see the first clinical trials opening for enrollment in the first half of 2022. And that's why it was so important to us today to start making this information really accessible at very, very easy to digest levels and have a Q&A with all of you. But we are looking at first trials in the first half of 2022 is what is expected.
Wow, that is coming quickly. And kind of on a follow-up to that question, do you think these clinical trials will be available for babies and toddlers or only for adults?
Yes, and I've seen that question coming up in the chat too. It's great to see so many familiar faces and names. One of the things that we do want to emphasize is, this will all be dependent on what each industry sponsor decides for their study. But what we fully expect at this time, because we are so focused and concerned about safety first, is that these trials will be focused on adults with CF first. This community has largely seen that happen as we've looked at modulator therapies that then moved down into pediatric ranges to look at safety of the same approved drugs in younger and younger populations, since those are still developing organs and developing tissues in younger humans. So we very much expect that as we are trying to look at safety first, we'll be looking at adults first.
Thank you. I think that makes a lot of sense. This next question is for you, Dr. Solomon. What are the current barriers to inserting the entire DNA code for normal or wild type CFTR into lung stem cells?
Okay, so that's a great question. So let me answer that question about the cells in general of the lung. The stem cells are cells that basically are very, very early precursor cells that can become any cells in the body. And so that stem cell insertion will be necessary for gene editing, especially for we call systemic, when there's whole body gene editing. We get to that point. We're not to that point yet because that technology is extremely complicated, both the editing and the stem cells. Okay, so we're not there biologically or scientifically yet. We will get there eventually. However, to get the cells to take up gene delivery in the lung, for instance, which will be the usual target for most of these initial therapies, we have to know a few things. One is we have to know how the vector and the gene therapy itself can penetrate the mucus layer that's remaining in the lung. So if it's very thick, it'd be hard for it to get through there. Then can it then target and hit and target the right cells? So we know there are lots of different cell types that line the lungs, for instance. One of those is a cell called ionocyte, which we think is this primary cell that expresses a lot of the CFTR protein. And so if we don't hit that cell, we might be missing the right cells that are getting what needs to happen in the lungs. And so that's another important thing, is knowing what cells need to be targeted, and can the vectors, in other words, the delivery methods for getting that gene in there, will they hit the right types of cells and appropriately deliver the gene to them? And the last is, how durable is the gene delivery? So does it last for a while, or does it sort of turn over as the cells turn over themselves? And so, for instance, could we give it to cells which are earlier in the progenitor phase of making lung cells, we call basal cells? Could we give the basal cells, which are kind of the precursor cells in the lungs themselves, to making the differentiated cells like the ionocytes and the cells with cilia and that kind of thing? Could we give it to them, and that would last longer? We don't know that yet. And so there's a lot of questions to answer. And so the first, kind of shots on goal, to borrow a common CFF term, will be, kind of thinking about the basic concepts and learning what we do about these initial therapies. But if we never start from somewhere, we'll never know how much we've overcome those barriers.
Thank you so much. We have a new question for Dr. Polineni. You mentioned how clinical trials are going to start in 2022, but the question is, how close are you to products for clinical use?
So that depends on a lot of factors, and much of that also really depends on what the results of these early phase studies look like. So we know that early phase studies tend to be shorter because essentially we're looking for a safety signal versus later phase 3 studies, where we're typically following people for a longer period of time to establish both the safety and the effectiveness. But in terms of when these are going to become approved for use, that really depends on the success of these early phase studies. So that involves so many factors, including the willingness of the community to enroll into these trials. So when there is active engagement and enrollment into the trials, they meet the sufficient numbers to be able to understand what's happening as an effect of the drug itself versus chance alone. So when industry sponsors decide how many people need to be in a trial or get to be in a trial, that's really based on statistical decisions. Even that is focused around safety, to enroll only as many people as necessary to find out precisely what is due to the drug itself versus by chance. So all of these types of factors, rapid enrollment into these studies, and then the effect and safety of the drugs themselves, those are going to be factors that lead to deciding when these will be available for clinical use.
May I amplify one point real quick, Deepika? Is that okay?
Yes.
I just want to say that, what we really need is we need an initial success. It doesn't have to be a home run, but it has to be at least a base hit, to borrow a baseball analogy. And if we have an initial success, like, for instance, the first drugs that treated F508 was Orkambi, not exactly a home run drug, but gosh, it was a success, understanding that we could use combination therapies to work on a complex mutation. If we'd never done that, we might never have gotten a Trikafta, right? So, we need those initial successes, even if there aren't the home runs. They get everyone in the community further engaged, and it shows us the blueprint to getting to some level of success, and we can build on that momentum. So we need that. That's why these first studies have to be at least a success in executing them, so we can get those initial kind of, hits and get runners on base, to keep borrowing that analogy.
I completely agree, Marty. And I also I know we've talked about this before, and I want to take it one step back further, and what we've also discussed together, that even ivacaftor that came before Orkambi, this is really, it can be kind of compared in what we're talking about today to like a proof of concept. So, many people were really excited when ivacaftor came out, but it was also really clear that this was affecting a much smaller proportion of the CF population. And yet we knew then what was possible. And as Marty says, then we started to see the development of therapy that reached more people and the safety of it, and we're able to keep building on that. In the same way today, if we start by looking at proof of concept in a narrow group of people where we're really trying to understand how to get these therapies to even work in these mutations and to understand the safety and effectiveness of them, we know that that's how we kind of build on the same building blocks that this community has helped to build in the same way with the story of modulators so far.
Thank you. That's very helpful in giving that context of the past and how it worked for Trikafta, I think makes a lot of sense. So the next question is for Dr. Solomon. Is there anything that is being worked on for people with 2 rare mutations?
Yes, the answer is yes. Work out of many labs and many clinical centers across the country, including my own center. We're developing new ways of looking at modulators for people with rare mutations, not to mention patients with rare mutations who are not modulator eligible will likely form the cornerstone of enrollment for many of the therapeutic trials we've just been looking at. So there are definitely many opportunities. Some of those are trying to gain the most access to people with modulators. I'm going to give an example. We're about to have enrollment starting in the first or second quarter of next year in a study looking at modulators for N1303K, the second most common of the class 2 mutations. And so we'll be enrolling nationally for that study. That's just one example that I'm very aware of because I'm involved with the study. But many other examples of those in the concept of theratyping and whatnot, many different ways to think about how can we get the most access to highly effective therapies across that spectrum that Dr. Polineni laid out so well for us earlier, and try to get those to people as early as possible.
Thank you.
Yep.
The question is shifting gears. It's going to be for Dr. Polineni. What are the mental consequences of genetic therapies for patients?
Well, that is a complex answer. So there's a number of ways to look at that question. So when we think about mental consequences for genetic therapies, someone may be asking that, and you're welcome to clarify in the chat if you sent that question, in terms of what the direct effects of a treatment may be on the brain. So what I want to emphasize first is that what we expect of these first trials coming around the corner is direct delivery into the lung. And the main challenges that we're talking about, as Dr. Solomon discussed, is getting those treatments into the exact cells in the lung where we need CFTR to function most. So from that, we do not expect a direct impact of a treatment or a drug itself onto the brain. But I think it is important to recognize that being enrolled in research, taking part in experimental research therapies, these can have their own mental health effects. It may be exciting, it may be anxiety provoking. We want you to make sure that you're talking with your team about that. We know that there's quite a bit of discussion in the CF community about potential mental health effects that people have reported on case-by-case basis or case series at this time in taking CFTR modulators. And we do know that CFTR is expressed in cells in the brain. And so we're really learning more as a community about what the impacts of correcting CFTR may be on the entire body. But the most important thing to take away from this discussion is that we can only learn about the complete effects of this as patients and their physicians report them. So at this point, at least in correcting CFTR through modulators, there has not been any kind of signal that has met a threshold that would raise concern at approval levels. But we really will continue to learn about the effects of correcting CFTR throughout the body as we continue to go.
Thank you so much. Sorry, I lost my place. So the next question is for Dr. Solomon. How far along is the research into whether CRISPR could possibly benefit those living with CF?
Okay, so let's make sure we understand what CRISPR is. So CRISPR is a derived enzyme that can actually perform gene editing. It's not a human-based enzyme, but it can be used in human cells for gene editing, and has actually been developed and is in human studies for certain diseases. One example is that is an ongoing NIH-sponsored study for sickle cell anemia, which is a condition which affects mostly African-American or African folks from that descent, and causes a severe anemia and can cause death and multiple other problems. And so there is a gene editing process that's being looked at for that condition. We're not there yet for CF, but the CRISPR thing is probably some years off. There's honing it to the lung and other organs that we need it to hone for CF is not there yet. For many of the reasons we talked about earlier, getting it into the right stem cells and whatnot for correction. That said, the technology is there, and so there will be a way to move that forward, but it is going to be a slow process compared to delivery of gene or mRNA that we've been talking about.
Thank you. I believe, Dr. Solomon, you were the one who talked previously about systemic delivery. We have a question about, do you see mRNA delivery or gene delivery potentially impacting other cells, like liver or pancreas? Is that what you're referring to?
Well, it depends on how the delivery. So most of the viral associated or the viral mediated will be delivered to one organ. There may be possibilities for a systemic administration of gene in different ways, the bloodstream and whatnot, but that will have to be a different sort of honing process, which is not reached, ready for patients. So most of what's ready for patient delivery will be local type of administration of these types of things, like to the lung or other organs, initially.
Thank you. There's a question maybe for Dr. Polineni. Could sweat tests become one of the annual tests to monitor CFTR function?
Can you ask the question again, Sylvia?
Oh, sorry. Yes. Could sweat tests become one of the annual tests to monitor CFTR function?
So again, that will really depend on systemic delivery versus lung delivery. So we've seen in the case of CFTR modulators, as they're being orally taken and absorbed throughout the body, that we can absolutely measure changes in sweat chloride as that's being taken up in tissues all over the body. But when we're talking about these types of therapies, and again, we are initially talking about lung delivery, we would not necessarily expect to see sweat chloride change. Dr. Solomon and I have actually talked about this when talking about potential other new trial designs. And although we are curious to then understand what may be systemically taken up as it's being delivered and taken up in the lungs and see if there is even any change or signal, it is not expected as a demonstration of effectiveness. However, it is a way that we will continue to learn more about the science of what's happening as we are working on these gene therapies and they're being delivered in the lung.
Thank you. I'll ask this question to Dr. Solomon. Let's see, or if Dr. Polineni, I'm not sure who can better answer it. Some people have not been able to take Trikafta because of the way it's processed through the liver and the way it affects the kidneys. Are any of the drugs that are being developed not processed through the liver?
I guess I can try to start with that question. So the locally delivered drugs, like for instance, if we take a nebulized viral delivered gene replacement or mRNA, it would go into the organ directly. So all of the metabolism would be through the cell itself, the cells it's being delivered to. So it's unlike a drug, because what happens with a systemic drug that goes through your gut and gets absorbed up in your bloodstream eventually through the gut is it has to go through all these different processes by which the body processes it and tags it as toxic or not toxic, and then clears it out after it's been used into what's called a metabolite or a drug metabolite. Well, that wouldn't happen the same way if you're delivering a drug locally. So there would be a little bit of that, but most of the removal of the drug would be through the cell turnover itself. So the cell would have the gene and all that stuff in it, and the cells would process that out. So it's a different process altogether. Now, when we got to systemic administration, let's say we got to the point where we were doing gene editing to the bloodstream for all over the body, that might have some effect in the liver and other processing organs like the liver and kidneys that do process drug metabolites and drug clearance and all that. And that may become important, but shouldn't be as important for a locally delivered drug like a nebulized therapy to the lung itself.
Thank you. We have a question related to kind of the pandemic, the COVID vaccine way they were developed. So did CF mRNA research help with COVID vaccine? And will the COVID vaccine help with CF mRNA? Dr. Polineni?
To my knowledge, and I welcome Dr. Solomon to also chime in, to my knowledge, I'm not aware of ways in which CF-focused mRNA research informed COVID vaccine development. However, the other way around, I do think is absolutely true, because we as an entire community and as an entire nation are learning about mRNA delivery and the overwhelming safety that we are generally seeing with the COVID vaccine to date in real time. So when we talk about the concept of mRNA delivery, it's really quite similar here in terms of what's being delivered. Essentially, we're talking about the mRNA for the CFTR that will lead to CFTR protein in a lipid envelope. And so each company has its own kind of proprietary envelope to have its own technological advantages to be taken up into the cell and have that released and working there. But as we've watched that kind of principle of seeing that affected through the mRNA vaccine with COVID-19, where really the mRNA was just for that spike protein, again, the lipid envelope taken up into the cell and released there. You know, we've really learned about what some of the side effects may feel like. Some of that, as we've learned from the data of these studies, may even just be related to the lipid portion itself and not necessarily the mRNA aspects. So yes, I do think that as a CF community, we absolutely are learning about the experience of mRNA research through the entire country's experience with the vaccine programs.
I was just going to make a brief comment that to amplify that, the Johnson & Johnson, the single shot vaccine, that is actually delivered by an adenovirus directly. That is the vector for that mRNA. So it's teaching us, as Dr. Polineni was saying, that we can safely use those types of vectors, including viral vectors, which has been the initial approach for delivering gene therapy going way back years ago. And so that it can be a safe approach for patients.
Yes, absolutely. Please pardon me. I'm speaking also from direct experience of vaccines.
Yeah.
Thank you both. This has been such an interesting conversation, and we have like our list of questions ongoing, but we're getting close to the time frame. So I'm going to end with one final question, all of us, and I'm going to answer that one too. But just to remind our audience, if you have more questions, research.cff.org is where you can submit them. And so our last question to end is, what are you most excited about for CF research in the coming year? And as a parent of a child with I'm going to start by saying Trikafta has been amazing, and these modulators for a lot of people with CF, but we're still missing some people. So I think my hope is that research leads to treatments or medications so that every single person with CF can have an improvement in their quality of life. Trikafta also still depends on people taking this medication every day. I really look forward to having a time where people with CF may have therapies that target the CF defect, and that will ease their treatment burden and give them better quality of life. So Dr. Polineni, do you want to go next?
Sure. Sylvia, first of all, thank you so much for an amazing job moderating this session today. And I think when I think about what's most exciting, it's really building on what you're saying. I think the three of us, as we've talked coming up to this session today, each have our own experiences with communities that have been marginalized, whether that is Hispanic communities that have not always been able to participate in research in the same way because of a lack of translation-informed consents, or as Marty has shared, you know, a family member of Jewish descent with a rare mutation, or for myself, just as a woman of color, understanding and looking at how health inequities impact health in rare disease. One of the things that I think is the most exciting that I know we're all focused on is that these therapies are really looking at people with any types of mutations in CF. And so it's an opportunity for people who have perhaps been more marginalized from trials in the past based on their mutations to be engaged in research that is focused towards helping people like them. And that is so important to me and to all of us. So I think that's what I'm the most excited about now.
I'll just amplify thanks to everyone for attending and for Sylvie for great moderation. And thank you for all your enthusiasm about this topic and, for moving this research forward. I guess I'm going to give a little bit more of a personal excitement rather than a generalized excitement because you guys have captured the kind of spirit of this, but I'm very excited. This year, we're going to be trialing a new device that will look directly at CFTR function in the lungs. And we think will be an awesome way of, at our center, I think will be an awesome way of detecting the delivery of these types of methods, and hopefully will become a useful tool for clinical trials. And we're happy the CF Foundation supporting that work. So.
Well, thank you, Dr. Polineni, Dr. Solomon. Thank you for everybody who attended. This has been very informative. And I just want to remind everybody that today's presentation was recorded. It will be posted on YouTube, and you will receive an email with a link to the recording. Thank you very much.